Hey guys. So today we're going to go over chapter 14, which is cardiovascular physiology. If you remember in anatomy or if you took anatomy and physiology, this is one of, I don't want to say it's really complex, but there's so many different moving parts to it. It can be a little difficult at times. So with that said, before I start on this, so I have to share with you a couple things that happened this weekend. So the first thing, my birthday was Saturday. So I have more additions to my pain cave, which is really awesome. So I got a functional trainer and a squat. I think they call it like a power cage. But anyways, super excited about that. But my favorite workout is the leg press. If you don't know what the leg press is, it's like this glorious machine where you lay on your back and then you push this weight away from you. So it's kind of like an inverted squat. So you're laying on your back instead of, and the angles and everything, it's completely different. Well, I had been wanting to do leg press for a long time, but since the gyms are closed down, I can't go to the gym. So I came up with this brilliant idea in one of my dreams. Or maybe I was like halfway awake. I was like, oh, I can just turn my couch in the basement into a leg press. And so I did. So I'm going to show you this real quick. So this is my homemade leg press. Don't look at the legs. I know they're kind of weird looking. So I've since changed it. But so this is leg press. Yes. So then I put my kids on there to increase the weight. And so that way I'm able to do this. And it makes me feel so happy. Like we're not in this crazy pandemic right now. So anyways, now I will go ahead and start on cardiovascular physiology after I tell you about this one last thing. Okay. So Saturday was my birthday. My dad ended up, so his dad died of a heart attack, a massive heart attack. His uncle last year, when I started teaching the cardiovascular system to my gross anatomy students, he ended up having having to have a quadruple bypass because he was like clogged everywhere in his coronary arteries. So that said, I get ready to teach about this cardiovascular physiology, and Saturday, my dad ended up going into the emergency room because he'd been working on his fence. He's a very big Russian, like he's got a lot of muscles and he's just really built. And so, but he hasn't really been working out very often. So he was outside working on his fence. He was throwing a sledgehammer around with posts and stuff. His heart rate went up to 170 and wouldn't come back down, even though he was just setting down. So they took him to the emergency room. And he had what's called AFib. And we'll talk about what AFib is in a little bit. But he had tachycardia, and the reason for that was because his heart was trying to compensate for the lack of arterial blood flow or the atrial blood flow into the ventricles. And so the ventricles were trying to keep up with the oxygen demand. And it was just turned bad. Anyways, he's fine now. Everything went back to normal. But yesterday they said that if he didn't come out of AFib, then they were going to have to put him out, shock him to try to get his conduction system to kick in going again. So it was the worst birthday present ever. So I told him he was in trouble, but we had already celebrated the day before. So anyways, all that said, that was like almost 4 minutes of rambling. Anyways, I would be talking about this in class, so this is kind of like we're in class. I'm still sharing stuff with you guys. Okay, so what you're going to learn in this, we're going to go over the cardiovascular system. What components are there in the cardiovascular system? We're going to talk about some of the pathology that happens with the cardiovascular system throughout this presentation. Some of the stuff I'm going to skip over because it's stuff that, unless you're in the field, you really don't need to know all the little tiny equations and all the little things that you're supposed to use to calculate different aspects. Anyways, you're going to learn about the pressure, flow, volume, resistance, and their effects on cardiac output. Also the cardiac muscle and the heart. And remember when we talked about depolarization, repolarization? Well, we're going to talk about that with the cardiac muscle cells as well as the nodal cells, which are part of the conduction system. They're the electrical cells that are sending the signal throughout the cells like, hey, we're about to contract. And then we're going to learn about the heart as a pump and taking everything into consideration, we're going to look at everything. We're going to look at the pressure within the ventricles. We're going to look at the pressure within the the aorta. We're going to look at the lub-dub sound, the EKG, all of it overlapping when we look at the cardiac cycle. So overview, the heart's going to be the pump, the blood is going to be the fluid, and then we have the blood vessels, including the capillaries, which are tubes. Now, each blood vessel is going to be different in their own respect, and we have a chapter, which is chapter 15, is going over the blood vessel. So we're going to get more into the different details about those. But when you get down to the capillary level, that is 1 cell thick. That's going to allow for rapid gas exchange through diffusion. The responsibilities for the cardiovascular system is to transport wastes, waters, hormones, nutrients. Did I say that? So it's going to send out a lot of things. It's going to move things like immune cells, antibodies to wherever they need to go. And then it's going to eliminate carbon dioxide as a byproduct, and also heat and metabolic waste. So that's all what is traveled. So the cardiovascular system is part of the waste removal system. So that's good. Blood vessels, we have arteries and veins. Now, hopefully, any artery is considered taking blood away from the heart. Now, we have 2 different circuits of circulation. We have the respiratory circulation. We have the systemic circulation. Not all arteries and not all veins are going to be oxygenated or deoxygenated. Any vessel that takes blood away from the heart is considered an artery. Any vessel that's bringing blood back to the heart is considered a vein. So we're going to get into the specifics, especially when we look at the separate systemic systems, separate circulatory systems, the respiratory as well as the systemic, completely different. So the heart's going to be divided into 4 chambers. You have a septum that's going to divide the left and the right, and then you're going to have another division from the atria and the ventricles. So the septum is called the interventricular septum because it's in the middle of the ventricles. And then we have the atrium, which is going to receive blood coming back to the heart. So it's going to be attached to veins. And then the ventricles are going to pump blood out of the heart. So those are going to be connected to arteries. So the pulmonary arteries, the aorta, which is an artery, the coronary arteries, which are the first vessels right after the left ventricle, after it leaves. You have the coronary arteries that are going to receive first dibs on the blood. And then you have the blood, which is going to contain cells, the red, the white, and also plasma or platelets. And then we also have the plasma. Now, just mentioned a bit ago about the pulmonary and systemic circulation. So the pulmonary arteries are going to be deoxygenated. Okay, so the right side of the heart is sending blood to the lungs to get reoxygenated. So the veins that are coming back to the heart into the right ventricle, that's deoxygenated. The pulmonary arteries that are taking blood to the lungs are also deoxygenated. Okay, so and then when we get to the left side, the pulmonary veins that are coming back are bringing oxygenated blood, which is different from when you had the veins bringing blood back to the heart, which were deoxygenated. So deoxygenated or oxygenated blood completely are out of the equation on this, but it's good to kind of understand. So the pulmonary veins are going to be oxygenated blood, bringing it back to the left ventricle the left atrium, and then into the left ventricle. So here are these are major vessels. That's part of the pulmonary system for circulation. Then we have the aorta, which is, it's an artery and it's taking blood away from the heart. It is oxygenated blood. The inferior and the superior vena cava, which are going to come right in here. And after this is all over, you come down to, even if we're not in session, whenever I am back at the cadaver lab, you are more than welcome to come down there. Because I really want you to see where the heart lies in the chest. It's like, and I'll talk about it, there's a part of the presentation where it shows where it lies, but it's right behind the sternum, and it's like right here. You can put your fist up to your chest and you can figure out where it actually lies. So that's one of those things. So whenever I'm back at the cadaver lab, you guys are more than welcome to come down to see where the heart lies. But you can see in the heart that there's the inferior vena cava, superior vena cava that are coming in and dumping blood back into the right atrium. So that's kind of just amazing. This is just a table that shows the transporting within the cardiovascular system. So this is the different substances that are moved, and this is going to or from. So you can review that. Now, when we look at the heart, so this is the heart, and then this is just the different circuits. So this is going to be the lungs, this is going to be the respiratory system, and then the rest of it is going to be the systemic system. Now, I want you to pay attention to the hepatic portal vein. This is where we're going to have multiple multiple capillary beds. One is for deoxygenation, but then there's another one for filtration. So here, the digestive tract, this is one capillary bed, and this is just picking up nutrients. And then you're going to go over to the liver, and it's going to filter the blood. So it's going to filter the blood, and it's also going to finish some deoxygenation. But just so you know that when you have a portal system, there's at least 2 capillary beds that are involved. Let's see. Oh, sorry. I wanted to stick here real quick. So here's the right side of the heart. Here's the left side of the heart. Now we're going to get into more details, so don't worry if you don't grasp it yet. If you do, that's great. So this is the right side. So all this blood is coming into the right atrium, then into the right ventricle. Then it's going to be pumped out to the pulmonary trunk, which you have pulmonary arteries because it's moving blood away from the heart. Now it is blue. Okay. Now other arteries like the aorta, this is oxygenated, so it's red. So there's a difference here. When you get into the respiratory circulation, this is a flip of the deoxygenation versus the oxygenation in reference to the veins and the arteries. So as long as you know that arteries take blood away from the heart, completely independent on if it's oxygenated or deoxygenated, then you'll definitely grasp that. So blood goes to the lungs, we get deoxygenation to reoxygenated blood, and now it's going to come back to the left atrium, to the left ventricle, then out into the aorta and back into circulation. So these are some of the different factors that are definitely going to have an effect on the type of blood pressure that we have and the cardiac output, basically. So the blood is going to move from an area of high concentration to an area of low concentration, very similar to any kind of passive movement that you have, diffusion, osmosis. So it's going from high to low. The highest concentration or the highest pressure that we have in the body is within the heart. So whenever the ventricles contract to push that blood from the left ventricle or the right ventricle, I'm just going to hang on to real quick, left ventricle and up into the aorta, that is under such high pressure, but along the rest of the circulatory system, it's under low pressure. So that's why when you have a higher pressure, that's going to push stuff down the pipe. So it's not that when the heart beats, everything is under that same pressure. It's not. It's originating from the heart and then diffusing throughout the body, or diffusing, making that pressure diffuse throughout the body in relative terms. So the pressure gradient is delta P, and there's going to be the difference in pressure between 2 different regions. So like at the aortic arch versus the renal veins, or renal arteries. So totally different. They're going to have 2 different types of pressure, but that delta P, or the pressure gradient, is going to give you that change or that difference within the number. Blood flow out of the heart, that's the highest pressure, and it's going to go into a closed loop that is under lower pressure. So it's just going to move that blood back along, and that's what's going to help to move that blood back to the heart, is that pressure that's moving throughout. So the fluid in motion has two components: the dynamic, which is the kinetic energy or the actual moving energy, and then we have the lateral, which is the hydrostatic pressure. Now that is just the fluid, and you can see down here, this is the pressure exerted by a fluid not in motion. So you have fluid already there. So this is going to be exerted in all directions. So the pressure of a fluid in motion decreases with distance. So the longer it has to travel, the lower it's going to get. So pressure is going to be lost due to friction as the blood moves past the vessels. So let's say the femoral artery in the leg, that's going to have a lower pressure than in the aorta. So if you can kind of sense that, when we have a heartbeat, that's going to move the blood down to the different areas, but it's going to lose pressure as it goes. So the driving pressure, should pay attention to this term, the driving pressure, this is when the blood leaves the heart to the vessels. And this is created with each heart contraction. So the driving force is the pressure from the heart to the vessels. So the vessels, they can either dilate or they can constrict. When the vessels dilate, we are going to drop in blood pressure because you're opening the diameter or widening the diameter of a vessel. And when it does that, you have less resistance, less laminar flow resistance. You're going to have less resistance and a higher laminar flow. So it's going to go straight through the middle. If the vessels constrict or get smaller, the pressure is going to increase. So imagine if you have a hose and you have the regular opening of the hose, water's coming out, and then what ends up happening is if you put your thumb over it, it starts to spray because it's under pressure. So now it's going to spray out. So that's kind of the effect that you can see in the blood vessels as well. So volume changes. When there's volume that changes, it's going to affect the blood pressure in the cardiovascular system. So when you have in the atria of the heart, especially the right atria, it detects stretching. So there are baroreceptors that are going to detect any kind of stretching that occurs within the atria. Now, because the atrial walls are so thin, any kind of stretching can damage them. So it's very sensitive to increase in the volume of blood. Now we have roughly around 5 liters of blood in the body as it is. So when we drink a lot of water, when we go and eat that Chinese food, and then we go, and then we get really thirsty, we're going to chug a lot of water. Well, what ends up happening is that water is going to increase. It's going to be circulating within the blood. It's going to be absorbed from the digestive tract. It's going to be coursing around in the blood, and then it's going to increase our blood volume until our kidneys work up the nerve to kind of dump some of that out. So when that is raised, it's going to affect the blood pressure as well. Okay, so let's see. Okay, we're going to move. So here, so the mean systolic pressure. Did that give nope, it didn't give them. Okay, so the mean blood pressure. So this is, there's an equation that you can use to find this. It's the difference between the 120 over 80, which is 40. But there's a there's a way to calculate this, and it's like a third of the highest blood pressure plus 80. So I believe that's the equation. Okay, so I just wanted to make sure I give you the equation because I don't see it anywhere. I took a moment to see if I could find it in here and I couldn't. So I wanted to just let you know that what they do here. So let me get this pen out and write it for you. I think, no. Well, that's weird. Why can't I write on this? That's so weird. I could the other day. Anyways, so, well, this is going to be irritating now. Okay, so how they got the 93 is they take the systolic pressure, which is 120. They subtract the diastolic, which is 80 from that, and then they times that by a third, or they divide it. Yeah, they times it by a third, and then they add that to the diastolic number, which is 180. I'm sorry, and it's 80. So that's where they get the 95. So depending on wherever you have the different types of pressure within those areas within each contraction, diastolic versus systolic. So that's where you'll find this this mean blood pressure. So you'll see here in the aorta, it's 93. This is the highest amount of pressure within the body. And as you go throughout the cardiovascular system, within the vessels, it's going to plummet dramatically because it's traveling a very long distance. One, it's going to get into some capillary beds. That's two. And once it gets into the capillary beds, and I'll show you next chapter, that once it gets into the capillary beds, there's a huge rushing out of a lot of the fluid within the capillaries, and it goes into the interstitial beds. So I will cover that when we get there. And then by the time it gets to the venules and the veins, in the veins, it's already really low pressure. And then as you increase the diameter of the vessels going back to the heart, because all these veins are draining into it, it's going to lose more pressure. So with the veins especially, it's important to have a system in place to prevent backflow, and that's where we have some valves. Again, something I'll talk about later. But this kind of shows you the difference in pressure. So this is static pressure. It's pressure that's being held in place because of a cork at the end of this whole series of pipes. So here, this is representing the heart, and this is representing all the different vessels that are farther down. And so as you get farther down, you're going to have less pressure. So there's going to be pressure that's lost as you go throughout. So yes, you have water in here, and as the water flows past, you have some resistance that it's met. But as you get farther away from the main source of the water, the pressure goes down. If that makes sense. And so as you're losing water here, you're losing pressure. So anyways, I hope that makes sense. Okay, so the flow through a tube depends on the pressure gradient. So again, this is still using this type of concept whenever you have an open or a closed system, but it's open to the point that, we'll say this is like the capillary beds, because we have a lot of fluid leaking out into the capillary beds. So these veins are going to lose pressure as they get farther away from the heart. So we're going to have, say this vessel here has 40 millimeters of mercury. The other side has 15 millimeters of mercury. And this is very, I'm going to talk more about this next chapter when we talk about the capillaries, because this is very similar to the capillary beds on the arterial side versus the venule side after they've had a massive rushing out of water, or serum into the interstitial spaces. So anyways, that's when you have this change, delta P, in the pressure. So we have some resistance that are going to oppose the flow. Poiseuille's law is one of those. This is one of those equations I'm not going to have you know, because I'm not going to have you do a lot of the calculations for that, but it's good to know this is one of those laws that are going to take into consideration the length of a tube, the viscosity or thickness, which is represented by N, and then we also have the radius. Now, when you, like what I was saying with the hose, when you increase the diameter, water is going to have less resistance. It's not going to hit the walls. It's not going to come into a lot of resistance, so the water's just going to come out. Now, on the flip side of that, if you put your thumb over the hole and reducing the radius or reducing the area that the water can actually come out, it's going to be under pressure. So you can see that Poiseuille's law is going to increase, or you're going to have a greater resistance whenever you decrease the diameter, make it smaller. So when you have resistance, because of the viscosity of the blood, the higher the viscosity of the blood, you're going to have higher resistance. The reason for that is you have more cells that are going to be hitting the walls of the adjacent of the vessel. So you're going to have, a lot of blood cells coursing through there. That's why blood doping is so dangerous, because as you have more red blood cells coursing around, it's going to increase the viscosity. Also, dehydration increases the viscosity of the blood. So the more viscous a fluid is, the more thick. I mean, just look at maple syrup versus molasses. Molasses, there's a lot of things in there, and it's very condensed. It's very viscous. It's very thick substance. But then maple syrup's like water almost. So true maple syrup, not just the stuff that's sugar laden. Okay. And again, we've talked about the vasoconstriction versus dilation. Now, all of this is going to be within this Poiseuille's law. So you have the resistance equals to 8 times the length times the viscosity divided by pi and the radius to the power of r. So that is a very fun equation, but this is all looking at the type of resistance that's going to be met within the vessel. Okay, so flow usually means flow rate, the volume of blood that passes a given point in the system per unit of time. The velocity of the flow has this wonderful equation where you have the velocity is equal to the flow rate times the cross-sectional area of a tube. So the distance is a fixed volume of blood that travels through at a given period of time. Then we have the mean arterial pressure, and this is the driving force of blood. So again, it's a pressure that's reserved within the arteries during heart relaxation. So you see pressure, you don't just find zero pressure unless you get into the veins on the very large diameter where you barely see any pressure. So in the arteries, we have a lot of pressure built up. And that's where we get this mean arterial pressure, and that's an inverse relationship with the cardiac output times the peripheral resistance that you see. Okay, so this is the physics of fluid flow. So this image is basically showing you, we're not going to get into a lot of the details over here, but I just want you to pay attention to this. So you have a main vessel, and it has 2 different ports coming off of it. One port is quite a bit smaller in radius, where you have this tube over here, which is double in size. It's 2 versus 1. Now, this pressure, or this port over here, actually is under greater amount of pressure than B, because it has half the resistance. Remember that analogy of the thumb over the hose, and it's building up pressure behind it. So the pressure, if you were able to measure it over here, would be quite a bit more than the pressure that you find in this region. So the cardiac muscle and the heart. So again, the heart's going to have 4 chambers. It's mainly made up of the myocardium. So there's 2 major types of cells within the heart that you need to be concerned about. One, the cells that are actually going to be doing the contraction, which are the cardiac muscle cells, and then the cells that are going to be initiating the contraction, which are the nodal cells. So when we look at the different 4 chambers, you're gonna have a right and left atria, which are super thin walls, and those are the upper chambers. And then you have the paired ventricles, which are lower. They're the lower chambers, and they have a thicker wall. Even one is thicker than the other, and I'll show you that in a second. The blood vessels are going to emerge from the base. So where you have the heart, the apex is going to be closer to the xiphoid process of the sternum. And then the base is going to be towards the top of the sternum, towards the clavicle. Right? Yeah, okay. So here's just a nice little pathway. Deoxygenated, they go to the vena cava, the right atrium, right ventricle, pulmonary trunk. The oxygenated are going to go from the pulmonary veins to the left atria, to the left ventricle, then out to the aorta. Now, the heart is situated, let me see. Okay, so the heart is situated right in this area, but it looks like there's nothing around. When in reality, you have a lung over here, you have another lung over here, and everything's kind of pushed together And you have this heart that's encased in this thin little sac called the pericardial sac. And that pericardial sac is anchored with all different types of fibrous connective tissue. It's anchored to the diaphragm. It's anchored to different parts. It's anchored to the posterior aspect of the aorta, behind the aorta. I mean, it's anchored so because it is beating all the time. So you don't want that thing jumping around. So there's a lot of fibrous attachments to the heart or to the pericardial sac, to the surrounding structures. And it's just fascinating. Like, it's so amazing. I mean, just to see this is just, oh. Okay, so here's the wonderful diaphragm here. You can see that this is lying on top of the diaphragm. So the heart, the apex of the heart, which is on the bottom, the base is going to be on the top. Now in the lungs, when we talk about them, they're flipped. So the apex is like protruding through vertebrae 1 or rib 1, not vertebrae 1, rib 1. And then the base is going to be down here, kind of sitting over the diaphragm, not touching the diaphragm, but sitting over it. Okay, so it's going to be situated behind the sternum, but it's deviated a little bit to the left. That's why the left lung is only going to have 2 lobes, which is really amazing. Okay, so then you can see over here the different circuits and stuff. This is more of a representation, more than this, that shows you. And this is a cutout of where the pericardial sac would be. Normally when you open a chest cavity, the only thing you see is one giant sac here, another sac. Well, you have a lung here and then a lung here. It used to be in its own sac. But that pericardial sac, I cut a little window so you can actually see what it looks like. And then you have this, these fibers just attaching it to anchoring it to the diaphragm so it doesn't bounce all over the place. You also see it's going to be attached to the aorta, the pulmonary trunk. I mean, it's attached so well. So that way we have some control. Let's see. This is just another section that you can see. Yeah, so you can just see here where you have the right atrium. So we're looking over and down, so you can see some of the bronchial tree over here. You can see the pulmonary arteries going in, and you can see the aorta, which is resected or segmented. Anyways, that's fun. Right atrium, left atrium over here, the left auricle of the left atrium, the right ventricle and the left ventricle. Now, the reason that it's shown this way is because the heart is kind of deviated and turned posteriorly, so you see more of the right ventricle. It's not like, let me see, where did it go? I guess I don't have it. Yeah, so you can see all of the right ventricle, all of the left ventricle here. But in a true sense, you can see more of the right ventricle in the anterior view and more of the left ventricle in the lateral posterior view. So if that makes any sense. Okay, so then we have this little fat-filled area where we have some of the arteries and veins running through, and this is called the interventricular sulcus. So because it's interventricular between the ventricles, and so you have this little fatty substance, and it's nice. It's a nice anchoring area for the vessels. Then you have the coronary sulcus, where you're going to have these little vessels running through and around. So you can see it over here. Oh, sorry. Oh. And then you have the superior vena cava, which you don't see down here, is going to be the inferior vena cava, the aorta, the pulmonary arteries, we call this the pulmonary trunk, and then it's going to go into the right and left pulmonary arteries. Here's the heart. You can see that the left side is going to have a thicker wall than the right side because this is going to have to generate more force to push it a greater distance, whereas the right side is only pumping it to the lungs, so it doesn't have to completely overpower the lungs. Otherwise, we'd be in a world of trouble. Looking at the myocardia, you can you can actually see how these cells are striated, but they have these wonderful little junctions here called the intercalated discs, where they have gap junctions. They also have desmosomes to anchor these things in place. This is also with the gap junctions is going to help to spread action potentials, nutrients from one cell to the next. So here's that septum I talked about, the interventricular septum. And this is going to separate the two ventricles. Now this is also going to serve for part of the electrical conduction to fit down into this sulcus, and then you have some Purkinje fibers that are going to come up on the sides, but we'll talk about that in just a little bit. You have two major sets of valves. You have the atrioventricular valves, and then you have the semilunar valves. The atrioventricular valves are going to divide up the chambers from the atria to the ventricles. They have they are tethered to the wall of the ventricles by way of the chordae tendineae. So those are going to be anchored on the papillary muscles of the heart wall. So whenever the heart is relaxed, they're pulling these little leaflets open. But when they contract, these papillary muscles move up closer. It gives slack on these, so the force within the ventricles actually slap these closed, so it can't backflow into the atria. So those are the atrioventricular valves. So you have the right one, which is also called the tricuspid valve, and then you have the left AV valve, also called the bicuspid valve, also called the mitral valve. So those are this one's going to have two major names, whereas this one only has one. Two. This has three. This has two. So the right AV valve, tricuspid valve, and then the left one has the left AV valve, the bicuspid, and the mitral valve. Okay, then we have the semilunar valve. So you see this semilunar valve, and we don't really see this one. It's kind of hiding back here. This is the respiratory semilunar valve, and then we have the pulmonary semilunar valve behind this. So they are going to work based on pressure. So just like how these worked on pressure whenever the ventricles were building up force, the papillary muscles went up, gave slack, and then slapped them closed. However, with the semilunar valves, whenever the pressure in the ventricles overcome the semilunar valves, it pushes the semilunar valves open. But as soon as the ventricles, they lose that pressure to overcome the force, the backflow of the blood are going to cause these semilunar valves to close. So then blood flow won't backflow into the ventricles. So I hope that makes sense. Two sets of valves: semilunar valves and AV valves. AV valves are operated by chordae tendineae, and then the pulmonary artery, or the semilunar valves, are working on pressure, backflow pressure, to close them. Anything else? It's okay. Okay. We just talked about these, so you can capture all that information here if you don't fully understand it. Here's that bicuspid valve, also known as the mitral valve. This is the one that's replaced a lot, so I don't know why it has the mitral. I don't know. I don't remember where that name comes from. There's a reason. But the coronary circulation supplies the blood to the heart, and those are the coronary arteries. So after the blood has been oxygenated, the coronary arteries are the first vessels that come off the aorta that are going to give the heart its blood. So it's going to give the blood back to the heart, and because the heart is pumping and it needs oxygen as much as possible, so they are going to get the blood first, and then the coronary arteries are going to take blood back. So this is, I kind of already gone through this because we talked about the valves. You can see an aerial view of the semilunar valves. No, these are the semilunar valves, and here's the AV valve, and here's another AV valve. So the tricuspid valve, bicuspid. Look at the tricuspid, you have 3 cusps. Mitral or bicuspid, you have 2 cusps, which that's where the bi comes from. And then we have the semilunar valves. Let's see. think I've covered all of that already. But again, you can use these to kind of, oh, actually, I'm going to show you this. Okay, so it's actually putting in motion what I was trying to explain. So when the heart is relaxed, you have the papillary muscles which are pulling on these chordae tendineae to open those AV valves. So it's pushing blood in. The flow or the blood pressure in the arteries where the semilunar valves are, it is higher than the ventricles, so it's keeping these semilunar valves closed. So this is a passive flow of blood into the ventricles, whereas here, when the ventricles are ready to contract, it is going to, I don't know what that sound was, but the ventricles are going to contract. When it contracts, the papillary muscles move up. It's going to close these cusps, and it's going to overpower the semilunar valves, pushing blood into the aorta. So if you want to go just back and forth between these two slides, that's going to really help you to understand the mechanisms behind these chordae tendineae and the papillary muscle and their role in the whole contraction. So the coronary circulation, so you have two coronary vessels that come off the heart. You have the right coronary artery and you have the left coronary artery. So this is the left coronary artery. Now the left coronary artery is going to give rise to two different vessels. You have the circumflex that's actually going to run the circumference, so that's an easy way to remember. But the more important one is that it's going to give rise to this wonderful vessel here called the anterior interventricular artery, also known as the anterior descending, or another name is the widowmaker, because this supplies blood to the left ventricle. If this gets occluded and there's no anastomoses to kind of bring blood into that area, an anastomosis is just another connection to another blood vessel. There are no anastomoses to the LAD, the left anterior descending. So you have this vessel. If it gets occluded, then you have a widow that's been made. Does that make sense? It's kind of brutal. Oh, so sad. So anyways, those are the two major arteries that come off the left. There's a lot of little tiny ones that even come down here. Like my uncle, he had see, there's some smaller anastomoses down here. But again, this blood's going to travel here and it's going to go over here. So anyways, and plus this is for the right ventricle. You don't have anything for the left ventricle, which is sad. What was I going to say? Oh yeah, my uncle. So he had this little peak here at the apex, this little tiny vessel here. It actually has a name, but unless you're going into the cardiology field, then this really wouldn't pertain to anything that you need to know. But so this little, one of the grafts didn't take for my uncle. So this didn't fully get blood supply. So there's a part of his heart that has been damaged, and it doesn't contract anymore. So there is dead tissue there that doesn't get blood anymore. And so that's kind of sad. In fact, so the whole reason that I'm even in this field, that I'm even teaching this, is because when my grandfather passed away from a massive heart attack 4 years ago. No, sorry, not 4 years ago. Gosh, it was when I was 16, actually. That was way more than 4 years ago. It was after a Chiefs Monday Night Football game. My dad took the trash out, and my dad and him were talking about the football game at Tamarick Vanover. If you guys remember Tamarick Vanover, he was fast, super fast. But anyways, in that particular game, the Monday Night Football game in October of 19-something, 1994, is that right? Anyways, they did, it might have been 1995, but he did this run that they went into overtime. The opposing team kicked. Tamarick Vanover caught it and ran it for an 85-yard touchdown. And so we won in overtime. So when my dad went up to talk to him, they were talking, they were chatting, and he was so happy. My grandfather was just so elated. He said something and he started laughing, and then he grabbed his chest. He was gone. He died. No bueno at all. So anyways, it was a very sad situation for him. And then my uncle had his thing, and then my dad had his. Anyway, let's move on. So we have the right coronary artery. So, and this is going to give rise to the posterior interventricular artery, which is right here, or it's also called the posterior descending, but really you just need posterior interventricular branch is fine. And then it also gives rise to the medial. Oh, it's not even on here. The median artery. So here's the median, which is right here, and this is not labeled, which makes me so sad because you guys should know this. But this one, I guess it's okay. But there's two major branches from this one, and you just don't get to see it. Oh well. So all of these are going to go around to the back of the heart, and they drain. Oh, actually, so those are the arteries. The veins are going to pick up. So here's the great cardiac vein. You have the middle cardiac vein, and you have the small cardiac vein. Now, the way to remember this. Now, you can look at the what is it called? The diameter to show you if it's great or small or middle. But the way that I tell my students to remember them is the great cardiac vein is on the left ventricle side. This has a greater responsibility at draining the blood from that heart because it's pumping to the rest of the body. It has a great responsibility. So this is the great cardiac vein. The small cardiac vein is going to be on the closest to the right atrium, which it has a small little amount of blood that comes into the atrium. And then on the back, we have the middle cardiac vein because it's running down the middle between the left and the right ventricle. So that's a way to remember it. And the small doesn't do that. It just rides up here next to the right atrium between the right and the left ventricle, or the right atria and ventricle. So it's just a small little diameter. But both the great and the middle are going to run in a inferior posterior fashion. So you can see that. Anything else? Not really. You can also see this part of the cardiac vein over here. Okay, so moving on. So the cardiac muscle cells contract without innervation. And the innervation means the sympathetic nervous system. I mean, it has an effect, but what causes the heart to contract are these cells here called the autorhythmic cells, also called the nodal cells, but this book doesn't use that. And so they have their own inbuilt ability to beat without anything telling them to beat. So yes, it can be influenced by the sympathetic nervous system and the parasympathetic, but but nothing tells it to go. It's autorhythmic. It's built in. It's autopilot. So this is the signal for the contractions. We have smaller and fewer contractile fibers compared to the contractile cells, and they do not have organized sarcomeres. So these are the pacemaker cells again. So make sure that you know that. The contractile cells, these are going to be striated fibers organized into sarcomeres. We made eggs yesterday for what is it? You know, you decorate eggs for Easter, and I drew a sarcomere on some eggs. I actually have a photo of that I should put up, but I won't. So cardiac muscle versus skeletal muscle. Again, you have the intercalated discs, which you have the gap junctions included. Cardiac muscle cells are smaller and they have a single nucleus per fiber. You remember, skeletal muscle does not. The cardiac muscle cells branch and they join neighboring cells with that intercalated disc. You have gap junctions for spreading nutrients and action potential. You have T-tubules, which are larger and branched, and the sarcoplasmic reticulum is smaller, and the mitochondria occupy 1/3 of the cell volume. That's pretty incredible. We don't use our quads all the time. We don't use our abs all the time. I mean, we don't use a lot of the muscles as much as we use the heart. So the heart has to have all these mitochondria. They have to have a more complex, more user-friendly apparatus within the cells, just so they can work the way that they're supposed to. So again, you can see this here. This does kind of look like a muscle cell, a skeletal muscle cell, only because of the overlapping contractile fibers. So you can see that here. You have the mitochondria, and you can see this is one of the cross sections of the intercalated discs. So one of the major things about the heart, when you are dealing with looking at the contraction versus when you're looking at the contraction of a cardiac muscle cell versus a skeletal muscle cell, is that the cardiac muscle cells really going to use calcium quite a bit more than the skeletal muscle cells. And yes, you use this calcium in skeletal muscle contraction because you have the troponin, and then you also have, the nerve impulse that you have calcium going through, pushing out the acetylcholine. However, calcium is used in a different fashion in the cardiac muscle cells, and that's to prolong the contraction to make sure that the heart doesn't lock up. So I'm going to go through that in a little bit. But again, we are going to have calcium binding to troponin. That's normal. But we are going to have quite a bit more of a player, or calcium has a lot more to do within the heart. So here's a T-tubule. Here is a calcium channel. You guys remember calcium channels? If you guys have been around anybody that's had high blood pressure, any heart arrhythmias, the calcium channel blocker is to stop the amount of calcium coming into the heart. So this is one of those, actually my dad, that's what he was sent home with. So calcium is going to come in. This is the sarcoplasmic reticulum where calcium is stored. So yes, the cardiovascular or the cardiac muscle cells are going to have those. And then when it's released, the calcium is going to spark by the signal, and this is going to cause contraction. So again, it causes a lot of contraction within the body. So the more calcium we have within the heart, the more we're going to have contractions. When we go into relaxation, the calcium goes back into the sarcoplasmic reticulum. So there's lots of different things that go on here. The sarcomere length is going to affect the force of the contraction. So the more cross bridges that you have, and actually when the sarcomere is stretched, it actually increases the force of the heart. Now we don't see the same thing in a skeletal muscle cell, because a skeletal muscle, when you stretch out those contractile fibers, you're going to be messing up the heads, the myosin heads. You lose that connection with the actin, but it's different in the heart. So when you have the sarcomere stretched and you have the optimal myosin head to actin ability to create that link, that's going to create a very forceful contraction. So when you have a lengthening or a stretching of the heart, you're going to have a forceful contraction. I hope that kind of makes sense. So if you see some jumping around of this, my screencast-o-matic is very finicky right now, so I don't know what's happening. Sometimes it cuts out, sometimes it doesn't. So I have to restart. So sorry about that. So moving on to the myocardial action potential. So this is going to be very different than what you see in the cardiac or the skeletal muscle. So I'm just going to take you here real quick. So here, this is number 4. So this is the resting membrane potential, which is -90, different than what you've seen before, which is -70. So this is the resting membrane potential. Then when we have depolarization, we have the sodium channels that are going to open. We have a huge influx of sodium super fast. And then at the peak, whenever we start to go to repolarization, we actually have this plateau that happens. So we have the sodium channels close, calcium's going to rush in, and calcium's going to hang out there for a little while to try to prolong the peak before you go into repolarization. So too, what we have here, the sodium channels open, the fast sodium channels close, trying to maintain that positive charge as long as possible. And then once we get to repolarization, we have the calcium channels closing, and then we have the potassium channels that are going to open to try to go back to resting membrane potential and get ready for the next action potential. So, and the slides, a couple slides before this, talk that out here, and that's totally fine. So again, here is the muscle contraction, the membrane potential. So this is the membrane potential, and this, so you can see here, the cardiac muscle fiber, the refractory period lasts as long as the entire muscle twitch. So this is the depolarization. So when you have the depolarization, that's when you're going to have the muscles contracting. So here's the muscle contracts. It's going to be at the peak right before it goes into rapid repolarization here. So remember, when you're overlapping the actual physical muscle contraction with the electrical changes, that's what's different. So that's why you have two different peaks here. This is the electrical changes within the cell. This is the actual force being generated. I hope you see the difference here. So here you can see in yellow the refractory period. So you have an entire refractory period. You have the entire length to make sure that we don't generate another muscle contraction before that one has been met. So this complete refractory period, we can't generate another action potential until we get down here. So that's when we can generate another one. So you can see this is a way to make sure that the heart doesn't lock up. So if we were able to generate another contraction here, if we move this one closer to over here, then we would be in a lot of trouble, because that's when we're not going to be able to have a relaxing moment. So that would be bad. Skeletal muscle is different. Here's the total refractory period. Here is the membrane potential that you can see. Here's the muscle contraction. Here's that tension, the refractory period, tetany versus complete tetany or incomplete tetany to complete maximum tension on the muscle fiber. So these are just some differences that you can see. Now in the nodal cells, the pacemaker, this has this nice little threshold. So here's the pacemaker potential. This is completely separate from the muscle cells. So you're going to see this for the actual cardiac muscle and this for the nodal cells. So we don't have a plateau with the nodal cells because they're autorhythmic. They're the ones that are trying to conduct business here, trying to make sure that we actually have the message for the muscle cells to contract. So we have not only the electrical changes within the nodal cells, we also have the electrical changes within the cell itself. The muscle cell, the cardiac muscle cell. So here you can see the pacemaker potential, then we have the action potential. During the pacemaker potential, we have sodium going in, calcium coming in, and then we have more calcium, a huge amount of calcium coming in, and then potassium going out, and then this goes back to normal. So we have, a lot of calcium channels that are opening, so you can see that as it goes up. Anyways, this is a nice little table that kind of reflects the differences between not only the skeletal muscle, but the contractile or the cardiac muscle cells, and then also the autorhythmic myocardium. So the heart is a pump. We have some, I know I'm going to go over an hour on this, and I'm so sorry. So the heart is a pump. So it has its own electrical conduction. This is sending the electrical signal that's going to coordinate the contractions. It uses this nodal system or a conduction system to do that. And so I'm going to go through what those are. Here is the nodal cells, the SA node, and there are nodal cells within here that are not shaped like the muscle branches. The cardiac muscle cells, which are branching. Now here is the SA node. The SA node is situated in the right atrium at the top. So it's in the top right atrium. Now the SA node is going to depolarize. Now remember, it's just sending an electrical conduction. It's not contracting, okay? So it's sending the electrical impulse into both the right and the left atrium, and it's saying, hey, we're getting ready to contract. So then when we're at 3, then we have the contraction of the atria. And then here at the AV node, which is at the bottom of the right atrium, that's where all the action potential congregates. So it's going to bottleneck right at the AV node. And then you'll see this little bundle branch. So this bundle branch is going to come out from the AV node and connecting it to the right and left bundle branches. So when you have the action potential spreading through there, you're going to come up on the wall of the heart where the Purkinje fibers are, and those Purkinje fibers are then going to tell the heart, hey, we're going to be ready to contract in just a moment. So this is the whole system. So it continues to do this the entire time. SA node throughout the right and left atria to the AV node. Why do they have that backwards? That's AV node. And then the AV bundle or bundle branch. And then we have the left and right bundle branches and then the Purkinje fibers. So know that in order. That's a really good test question. Put in this order or whatever. What else? Oh, right. So what I wanted to say, so we have the SA node. We already know that it fires at will. So inherently, it can fire at 100 beats per minute. But because we have the parasympathetic division having its tone or its innervating the heart, it actually tones it down to about 60 to 80 beats per minute. If we didn't have the parasympathetic innervation at all, we would be beating around 100 beats per minute. So the SA node can fire about 80 to 100 beats per minute. The AV node can actually fire, 50 to 70 beats per minute. Then we have the AV node, the bundle branch, which can fire about 30 to 50. And then we have the Purkinje fibers, the right and left bundle branch. Now, those cannot fire at a rate that would preserve life. So you've heard people having to have pacemakers. That's when this guy fails. When this guy fails, you have the backup system as the AV node. And I was explaining this to my mom yesterday. For some reason, my dad's AV node or SA node wasn't working like it was supposed to. It wasn't firing and it wasn't causing the atria to contract. So that was a big problem. So, so the SA node failed. For some reason, we don't know. It's back to normal now. But the SA node, if it fails, we have a backup system. So we have the AV node to get us through until we have some sort of intervention, medical intervention by either drugs or surgery. So kind of whatever happens there. So anyways, these two parts of the conduction system can actually sustain life for us. So that's wonderful. Sorry, I'm wearing a hoodie and my hair gets caught in weird places. So so the SA node and the AV node, they are, they beat enough to sustain life, but the other ones, they don't. So if our SA node fails, we have our AV node until we get to the hospital, which is really nice. And then if we need a pacemaker, in fact, one of my cadavers, they have a pacemaker. And so when you come to the lab, you can actually see it's underneath the skin, at least it was. And then it was, put into some of the veins and then it goes straight into the right atrium. So it's kind of interesting. So moving on. So an electrocardiogram, this is showing some of the electrical activity. Now, a lot of people think, oh, well, the EKG is only denoting, the contractions of the heart. That is not true. This is only the changes in the electrical conduction of the heart. So looking at this one, so we have this peak, we have this peak, and we have this peak. Very simply, this is the change in the electrical conduction of the atria when, like right before they go into contraction. So this is when the atria depolarize, okay? When you have the QRS complex, or I'll just say this peak until we get into the nitty gritty of it. This is when the ventricles depolarize. So this is the electrical change when the ventricles depolarize, not when they contract. If they haven't contracted yet, usually contraction happens in the peak. So here in the peak, that's when the atria contract. Here at the peak here is where the ventricles contract. But it's just denoting the electrical changes caused by that. And then we have this little peak over here, which is it's calculating the electrical change from when the ventricles go into relaxation or repolarization. So the ventricles, you can see the depolarization, the repolarization. The atria, you can see the depolarization, but you can't see the repolarization because that's occurring the same time the ventricles are going through depolarization, and it completely masks the atrial repolarization. So you don't see that very well. Going back, did I miss anything important? I mean, this kind of just shows you all the different things. So P waves, the depolarization of the atria, QRS complex, ventricle depolarization, T wave, repolarizing of the ventricles. Now the atrial repolarization is within this QRS complex. The two segments, which you have the P and the R, which is the AV nodal delay. And then we have the T to P, and that's the ventricle and atrial repolarization or relaxation. So you have some other ones. Yeah. Okay, so moving forward, I'm not really going to go over this because I'm already pretty short on time. Now, when you have an EKG, it's really easy to read. You just want to make sure you have the 3 peaks. Okay, so you have the 3 peaks here. When you go over here, this is when you have multiple P waves and then an occasional QRS complex. What that means, it's a 3rd degree block. It means that the atria are firing more often than the ventricles, and they need to find out why. So then we have the atrial fibrillation, which is what my dad had. And you can see that you have the QRS complex, T wave, QRS complex, T wave, QRS complex, T wave. There's no P wave. He had no contraction or no depolarization of his atria. So he went into AFib. Ventricle fibrillation, VFib, is deadly because when you have fibrillation, it's basically a quivering muscle. It's just kind of contracting and just kind of moving. It doesn't do anything. So that's not very fun. So in this image here, what do you think this would be? Now just take a moment, you can pause it, and I just want you to think about this. What does this look like to you? Okay, so hopefully you paused that and you kind of figured it out. It is the 3rd degree block because you have more P waves than QRS complex. So you have P wave, QRS complex, T wave, P wave, P wave, P wave, QRS complex, T wave. So that is very similar to this. You might not have a complete identical you might not have a complete diving down or change in the electrical conduction that goes into the negatives, but you do, not in the negatives, but you have just a dramatic change here. And you might not see that when you look at somebody with an EKG. So, and this doesn't mean anything. It's just, it's just different. Okay. So you can go through this a little bit more in depth. When the nodal cells are sending out the signal that they're getting ready to contract, this is when we have the P wave. And then at the peak of the P wave, that's when we actually have the contraction. When we have the delivery of the bundle branches, so the message going through the bundle branches, that's when we're going to have a dipping of the wave. Then we have a QR, which is when we have the depolarization of the, not depolarization, but the depolarization. We're sending the message through the Purkinje fibers that we're getting ready to contract. And then at the peak of this contraction, that's when we're going to see the ventricles contract. So you can see here, this is showing the atria contract past the peak. Each textbook is going to be different. I've always taught, and it's always been taught, that right at the peak, that's when you have the contraction occurring. And this actually starts right after the Q, but technically it starts right at the peak of the R. So just wanted to point that out. When you go into ventricle repolarization, that's when you're right at the peak of the T. So anyways, and then that's the cardiac cycle. So the lub-dub sounds that you hear, those are going to be based on the AV valves and the semilunar valves. So the first sound is the lub sound. It's not as dominating as the as the dub sound. So the AV valves, when they close, they make the lub sound. It's a nice little lub. And then the dub, which is super dominant, that's going to be when the pressure from the arteries push back on those semilunar valves. They're going to cause it to close. So anyway, so those are the different things that are just kind of, again, it's just going into a lot of detail about the cardiac cycle. So I want to show you this. Now we have some key terms here, the end diastolic volume and the end systolic volume. So you're going to start on A. Now I want you to think about this, take some time, really digest this. If you're going into the healthcare field, you're going to need to know this anyways. So go ahead and just take the time now, get familiar with it. So the left ventricular volume. So this is the volume of the left ventricle. This is the left ventricular pressure. So you can see it goes up to 120. Here's 80. So when the ventricles are in relaxation mode, they're passively filling with the blood from the atria. So this is coming in. You can see the pressure is low. So the pressure, you can see the pressure is super low over here. We have the filling up of the ventricles because the volume is going up, but the pressure is low. And then here is the end diastolic volume. So that's the end number, or the volume of blood that's in the heart when it's in, what is it called? When it's right before it is about to contract. So that's when it's in its relaxation. So there's a little bit of pressure that comes up because that's the amount of force that the atria are generating to push that blood into the end diastolic, into the ventricle, into the left ventricle. So here we go. We have the maximum amount of volume, which is 130 milliliters, and then it is going to start generating a lot of pressure. Now, this isn't from the atria. No, this is going to be from the contraction that's about to happen. So we have a lot of pressure that's being built up. We're going to start putting a lot of pressure on this volume. And then at this point, this is the amount of force that's generated, or the amount of pressure that's generating when the valves are closed. Okay, as we reach 80, then we're going to be able to overcome the semilunar valve, the aortic semilunar valve, and allow that blood to flow into the aorta. So when we start to see this volume go down and the pressure continue to rise, that means the ventricle is contracting and has pressure to overcome the blood flow the blood in the aorta. So it's pushing that blood into the aorta. So that's why the pressure is still going up, but the volume is going down. So when you get over here, this is the end systolic volume. So this is the amount of blood that you have left in the heart at the end of a contraction. So when you squeeze blood out of the heart, it's not that all of it's going to go. Not all of the blood is going to leave the heart. There's going to be some that stays. And so in this case, 65 milliliters of the blood stays, and it's going to go through this cardiac cycle again. So this is one cardiac cycle where it goes through. I want you to take time and really digest that and really start to think about that. This is a very complex system. So the more that you study, it'll be better. Now this, called the Wigger's diagram, and this is very fun. There's a lot of stuff going on here, but I just wanted to point out some specifics. So here, in this top graph is the EKG, or ECG, however you want to say it. So you have the P wave, the QRS complex, the T wave. Here is a graph noting the pressure. So you have pressure in the left atria, you have pressure in the left ventricle, you have the aorta. So in the left ventricle is what you're going to see the most dramatic change in the pressure. So the left ventricle has the ability to go from, what is this, 40? No, sorry. What would that be? Like 10 millimeters of mercury, all the way up to 120. So when it's in relaxation, it's pretty low. Look at the aorta. So the aorta is about 90 millimeters of mercury, and it goes down a little bit. But then when the pressure within the ventricle starts to go up, because here is when we had the ventricles depolarized. You see the peak here. You can see all of this is denoted down here. So we're in ventricular systole. The ventricle is starting to contract, creating a lot of pressure. Here at this point is that pressure has overcome the aorta. And so now the aortic pressure is going to match that of the left ventricle, because it's the pressure that's being pushed into the aorta is the same as in the left, because the left's creating it. So then when the ventricle is done contracting, it's kind of petered out. It's, maximum contraction. Now it's going to start to lose pressure. So as the ventricle starts to lose pressure, the blood that's inside the vessel, the aorta, it's going to try to fall back into the ventricle, but it's caught by the semilunar valves, and it's going to balloon open, and it's going to stop the blood going back into the left ventricle. And when it does that, it's going to lose a little bit of pressure. And so that's when it's like, oh, it's going to fall back. And so it's going to lose pressure. That's what we call the dicrotic notch. You have a little drop in pressure because of the ballooning out of the aorta, or the aortic semilunar valves. Then it's going to reestablish itself. It's going to come up a little bit as the blood continues to pull back on those semilunar valves, and then it's going to continuously decline like we saw over here, and we're going to have another cycle going. The left atrial pressure, there's really not a whole lot generated except for a little bit during the peak of contraction, when they sent that little extra squeeze of blood into the ventricles. That's basically what's going to happen there. So here is the first sound, the lub sound. And that's going to be when the AV valves close. And those close because, remember, the ventricle's going up, that the chordae tendineae are becoming lax, and the pressure is going to slap those semilunar valves closed. So that's what's going to generate the lub sound. And so you have the semilunar valves open. And then over here, when they close because of that backflow of the blood, it's going to create that second sound, which is the dub sound. Now there's two other sounds, the cardiac sound 3 and 4. That's, you can't really hear unless you have cardiac 3 stethoscope. At least that's what it was back in the day. And then this just kind of goes through the ventricular volume, how much blood is in there. So it goes from, 135 all the way down to 65. So that's the amount of blood that's going to go in and out of the heart at any time. So please take some time, go through this. This is a fascinating system. It's just really amazing. Cardiac output, this is going to be the heart rate times the stroke volume. And if you can do some math over here, we'll just say that's what, 70 milliliters. The average heartbeat is 70 beats per minute, because it's 60 to 80. 70 is considered average. So that's approximately 4.9 liters of blood. So we're actually circulating within 1 minute. We're circulating 5 liters of blood in our heart at rest. So, and how many liters of blood do we have? 5 liters of blood. So your heart is circulating all of your blood within 1 minute. That's a pretty darn good job that it has. So, what else? So these are some of the controls that are going to have an influence of the over the heart. Parasympathetic is going to oh, that's a huge typo. Parasympathetic control is actually going to decrease the heart rate. So that's an important part. And the sympathetic control is going to increase the heart rate. So make sure you don't pay attention to this, because that is completely, when you're stressed out or when you're running, the sympathetic control is actually going to increase the heart rate in any situation. So whether you're running from an axe murderer or you're stressed out about something, you have, a pretty high heart rate. So with the parasympathetic control, it's going to decrease. So I'm just going to leave it at that. So usually the tonic control is the normal heart rate that we have, the average, the 70 beats per minute. That's maintained by the parasympathetic because it slows our heart rate down. So that's kind of important. So here is the parasympathetic neuron. And this is going to slow our heart rate down. So see, it even says it correct here. It decreases the heart rate, where here it says it increases it. Simple checking. And then the sympathetic neuron, this is going to increase the heart rate. Here are some different flow charts that you can look at this. When you have the sympathetic neurons, we have the beta 1 receptors, which are on the autorhythmic cells. And this is going to cause an increase in the sodium, increase in the in the calcium influx. That's when we have the beta 1 receptors activated. So you have activation of those receptors on the surface of the nodal cells. And so when you have that, you're going to have an increased rate of depolarization, increase in heart rate. On the flip side of that, we have the parasympathetic, which is going to decrease the heart rate by having an influx of the potassium and a lower amount of calcium coming into the heart. So there's some other controls on here. Oh, okay. So this is one that I wanted to go through. So any chemical that's going to have an effect on the contractile ability of the heart, we call it an inotropic agent. So, and it's called the inotropic effect. So we have positive inotropics and negative inotropics. Positive inotropics are going to increase in the contractility. So that would be like your epinephrine, your norepinephrine, digitalis. And then your negative inotropic agents are going to decrease the contractility, and those would be like your calcium blockers, things like that. So I think that is basically all that I have for you guys. So I try to keep it, I still went over almost at the same time of last chapter, but that's okay. So anyways, if you have any questions, I'm going to be Zooming with you guys here in a little while. Anyways, I'll have this up so you guys will have this for your evening. And I really hope that you guys are actually like going through these. That would be super helpful, because I'm going to start putting some extra credit in there somewhere so I can really tell if you guys are paying attention. Okay, thanks guys.